Hello student, good morning. To continue the lecture, that is, we have to study here morphology of Taenia solium, and remaining part, that is the life cycle of Taenia solium. Life cycle. Taenia solium or tapeworms are the hermaphrodite. Both the reproductive system present in the same individual. Taenia is a hermaphrodite. Reproductive organs are usually found in the proglottids. After 200th partition, some anterior proglottids are functionally male. Middle proglottids are both male as well as female proglottids. At the posterior end are filled with enlarged uteri. Structure of the proglottids in the middle region is as follows. Male reproductive system. The male reproductive system consists of the following organs. Testes. They are numerous small rounded bodies situated in the medullary part of the parenchyma. Their number is about 15 to 200 and are distributed throughout close to the dorsal surface of the proglottids. Some workers are of the opinion that the numerous rounded follicles in fact constitute a single and highly subdivided testis. However, the number of testes is of taxonomic value in the tapeworm. Apex efferent ducts. From each testis arises a fine ductule or capillary known as vas efferens. All the vasa efferentia unite to form a vas deferens. Vas deferens. It is a long tube arising from the middle point of the proglottid. It runs transversely to the left or right to open into serous. In some cystodes, the vas deferens enlarges to form the seminal vesicle for the storage of sperm. But in Taenia, no such enlargement is seen. Serous or penis. The distal end of vas deferens is modified into the protrusible serous. The serous is armed with bristles, hooks, or spines. It is irreversible through a male genital pore into a cup-like genital atrium of the genital papillae. Serous sac. Serous is surrounded by kind of muscular sac known as the serous sac. Its function is to erect penis during the copulation. Female reproductive system. The female reproductive system consists of ovary. A single bilobed ovary lies ventrally in the posterior part of the proglottids. Each lobe of ovary is dorsoventrally flattened and consists of a number of radially arranged follicles. The right and left lobes are connected medially by a transverse bridge, the ovarian isthmus, oviduct. The oviduct arises from the middle of the bridge. It is short but wide duct and joins the ootype. It is small round chambers developed at the junction of oviduct with the vitelline duct. It is surrounded by numerous unicellular Mehlis glands, the cells of which form an amorphous complex uterus. A blind sac-like or tube-like uterus arises from the ootype and runs forward in the segments. In gravid proglottids, the uterus enlarges and gets branched to occupy the whole of proglottids is remain filled with the fertilization over developing embryos. Vagina. The narrow tubular vagina arises from the receptaculum seminalis, runs obliquely to outwards to open into the genital chambers by female reproductive opening. The seminal receptacle or receptaculum seminalis is a small white tube connected on one hand with the ootype by a narrow spermatic duct and on the other with vagina. Vitelline glands. The vitelline glands is a compact elliptical mass of the numerous follicles situated posterior to the ovary. A single short median vitelline duct arises from the glands and penis into the ootype. The secretion of the vitelline ducts is rich in yolk and form covering of yolk and cell around the fertilization eggs. Mealy's glands, these are numerous minute unicellular glands situated around the ootype, emptying into its functions of the section of Mealy's glands is to case passage celled eggs into the uterus. Life cycle. Fertilization. Fertilization occurs by introducing the serous into the vagina of the same proglottids, and sperms are stored in the receptaculum seminalis, which later on fuse with eggs in uterus. Thus, in Taenia, self-fertilization takes place. Sometimes the anterior mature proglottids devoid of female genitalia can enter the stage of copulation only with the posterior minute proglottids with fully developed female genital organs. Fertilization following copulation between two different proglottids is sometimes termed cross fertilization to distinguish it from that occurring between gametes of the same proglottids. Self-fertilization. Branch of uterus, main stem, serous, gonopore, sperm duct. Thus the eggs are fertilized in the oviduct and get surrounded with yolk and egg cell in the ootype. These materials are provided by vitelline glands. The capsule eggs enters the uterus and are collected there. The uterus enlarges in size, gets branched and occupies the whole space. The eggs are small, measuring 40 micron in diameter. Cleavage. The zygote or egg cell undergoes cleavage. When the egg capable inside the uterus, the first unequal division results the large mesenteric and smaller embryonic cell. The megameres divides further from similar, while the embryonic cell divides, producing type of embryonic cells, large mesomeres and smaller micromeres. Thus, three types of the cells result from the zygote: small micromeres, medium mesomeres and large megameres. First larvae, that is the oncosphere. The repeated division micromeres or solid ball cells. The morula is formed. The megameres divided into redivide and becomes filled up with the yolk from the yolk cells and surrounded the embryonic cells just like in the envelope. It provided nourishment to the embryonic cells and finally disintegrates. The mesomeres form the inner thick striated muscles known as embryophore. Beneath the embryophore lies basement membrane. The morula develops three pairs of chitinous hooks. These hooks are secreted by the differentiated cells known as oncoblast. Now the six-hooked embryo is called as a hexacanth. It possesses pair of large penetration glands, is surrounded by the membrane of hexacanth embryo. hexacanth embryo. This stage of development is found in proglottids at the extreme posterior end of the body. Teats are liberated from the body 4 to 5 at times, passes quickly in the feces. Infection of primary host. The bladder worm Cysticercus is eaten with the pork by man. The bladders is digested and the young scolex attacks itself to the wall of intestinal by hooks and suckers. A rigid proliferation of neck is formed which develops proglottids of the adult tapeworm, while the anchoring itself to the wall of small intestine immature in 2 or 3 months. Pathogenesis. Both adults as well as Cysticercus stage are harmful. Adult worm. The scolex of adult worm remains embedded in the mucous lining of the alimentary canal. For the attachment, the injury of mucus may lead to bacterial infection. There may be abdominal discomfort, diarrhea, frequent hunger pains, nervousness, insomnia, nausea, and epileptica, etc. These symptoms are due to presence of the adult worms, are collectively known as teniasis. Cysticercus larva. Sometimes man may be accidentally ingest eggs or proglottids with the contaminated food. Seriously effects may be results from the cysticercosis in human. Treatment. Niclosamide is the drug of choice for intestinal infections. It is highly effective and well tolerated. Carbon tetrachloride and oleoresin are also used as an anthelmic for removed tapeworms. Dose of 0.4 gram each of atropine and acronyl or 1 gram of hexyresorcinol with 2 gram of acacia may be given. Cysticercosis is best treated in human by surgical removal of cyst when possible. If surgery is not possible, mebendazole can be administered. Disease. The infestation caused due to this worm is followed by abdominal pain, nausea, anemia, indigestion, diarrhea, eosinophilia, etc. The complicated disorders are caused due to the toxins produced by the Taenia solium. Generally, one pork tapeworm parasites is single host at a time. Prevention and controls. The prevention needs the ingestion of properly cooked pork. The teniasis can be controlled by the quinacrine and hydrochloride and atabrine. This is the life cycle of Taenia solium. Okay, thank you.